Welcome to Introduction and Food Safety for Chemistry 125 Edible Experiments. I'm Dr. Jay Ratliff. Let's get started. Each week, you'll have a variety of assignments due at the end of the week, that is, due at 11:59 p.m. on Sunday at the end of the week. First, you should read through the textbook pages outlined in the Readings and Quiz Questions PDF that I've posted on Blackboard. You should also read any additional documents that I've posted on that week's tab on Blackboard. Each week, there will also be a quiz due. For this first week, you should then also sign and upload to Blackboard the syllabus and food safety contracts. As a reminder, these contracts can be found at the end of the syllabus. As a reminder, throughout the course you must complete at least 5 out of the 13 possible experiments. The report for each experiment is due at 11:59 p.m. on Sunday of the week that they're assigned. For example, if you were to complete an experiment from week 3, that report is due at the end of week 3. If you were to submit it during or after week 4, it would then be subject to the late penalty outlined in the syllabus. Now, I want you to take a minute to pause the video and think about what exactly chemistry has to do with the food that we consume. Why is this a chemistry class rather than, say, a biology class? Remember that biology is the study of life, and chemistry is the study of atoms and molecules and how those atoms and molecules react with one another. Now, we are alive, and therefore biology is a large portion of what makes us what and who we are, but we are also made up of atoms and molecules. So any sufficiently detailed study of humans will ultimately have to examine how those atoms and molecules interact with one another. Now, most of your body is made up of just 4 elements: oxygen, carbon, hydrogen, and nitrogen. On the left side of the slide, you can see a variety of elements and their percent abundance in your body. The most abundant element by mass is oxygen. Now, this is because roughly 70% of your body is water. Of course, we know that water has a chemical formula of H2O. So each water molecule has 2 hydrogen atoms and 1 oxygen atom. Now, the oxygen atom weighs 16 times as much as a hydrogen atom. And because there are 2 hydrogen atoms per water molecule, that means there's an 8-to-1 ratio of oxygen to hydrogen by mass due to the water. Now, you might notice that the abundance of oxygen at 65% of your body mass isn't 8 times as high as the 10% from hydrogen. This is because both the oxygen and the hydrogen are also present in lots of other molecules inside our bodies, such as in proteins, carbohydrates, and fats. In these, hydrogen is present, on average, at significantly higher ratios compared to oxygen than the 2-to-1 ratio that we find in water. We can see other elements such as phosphorus, calcium, sodium, potassium, and others are also present in our bodies, although in smaller amounts. So now I want you to pause the video and think about why we eat. What do our bodies get out of the food and drink that we consume? Note that there are multiple answers to this question, so pause the video until you've thought of at least 2 different reasons. The biggest reason why we eat is that our bodies need energy, and we need lots of it. We need energy to be able to move our muscles and to run the various processes occurring in our organs. That energy comes stored as chemical energy in 2 different ways. Energy is stored for the short term in the form of carbohydrates like sugars and starches, while energy is stored for longer term in the form of fats. Our bodies use carbohydrates to store energy for the short term, minutes to hours. If we haven't used that energy after a few hours, our bodies convert those carbohydrates into fats for longer term storage. When we later need to use that energy, that is, when our bodies later run out of the carbohydrates in our bloodstream, we convert those fats back into carbohydrates for use. Of course, I'm glossing over many of the details of our metabolic processes here, but for now, I want you to remember that fats and carbohydrates store energy for us to use later. Now, in addition to energy, our bodies also need the atoms and molecules that make up our bodies in order for us to grow. The majority of the structure and cellular machinery of our cells are made up of proteins. So we either need to eat those proteins, or we need to build those proteins out of smaller building blocks called amino acids that are present in the foods that we eat. We also need a variety of other vitamins and minerals, such as calcium for our bones, sodium and potassium ions for our nervous system, and so on. So our bodies get all of these various types of atoms and molecules, as well as the energy that we need through the food that we eat. Now, atoms are tiny. So tiny, in fact, that not only can we not see individual atoms with the naked eye, we can't even see them with the best optical microscopes. Now, this isn't a limitation of our current microscope technology, but rather with the underlying physics. That means that no matter how good our technology becomes, we will never be able to use visible light to see individual atoms. On the left side of this slide is an analogy showing the relationships between the size of an atom, a penny, and the moon. A penny is roughly as many times larger than an atom as the moon is larger than the penny. So again, atoms and molecules are ridiculously tiny. So tiny, in fact, that there are more water molecules in a single cup of water than there are cups of water in all the oceans, lakes, and rivers on Earth. Now, in this class, we're generally going to be concerned more with general shapes of molecules than their exact structures. So I might show you an image like the one on the left, showing water molecules with two white spheres representing the two hydrogen atoms and one red sphere representing the oxygen atom. But we're generally going to be more concerned with a broad overview in how they interact with other molecules. So while you should recognize that their individual structures are more complicated, we are instead going to treat them as a good bit simpler than they really are. Similarly, I might show you an image like the blue circles on the right in order to represent those water molecules interacting with one another, and you should recognize that it's a fairly simplistic view that glosses over many of the details. Where I might show you the structures of a variety of molecules, I don't expect you to memorize those structures. Instead, I want you to focus on how the properties of those molecules cause them to interact with regards to our food. That is, when they're cooked, when we taste or smell them, or when they enter our bodies. Now, because our food is a collection of molecules, the properties of those molecules will determine the properties of our food. As we change the composition of the molecules in our food, we'll change the way our food looks, tastes, smells, and how it behaves. For example, the molecules present in pancake batter are different than the molecules present in a cooked pancake. It's this difference that causes the transformation of the pancake batter from a pale, runny, not so tasty, potential health hazard into a deliciously brown, semi-solid, yummy, safe to eat breakfast treat. So let's start by examining one very common ingredient in our food, eggs. Now, rather than starting by talking about how we use eggs in food, let's first talk about what the purpose of the egg is in nature. The hen lays the egg not to provide another animal with food, but rather to reproduce. That is, the egg evolved as a place for the chicken embryo to grow from a single cell into a chick that then hatches by breaking out of the egg. So each part of the egg evolved to serve a purpose in helping that embryo develop into a chick. On the outside of the egg, of course, is the shell that provides the physical structure that keeps the egg intact as it is developing. Now, you might think that the shell keeps all the molecules on the interior inside the egg and all the molecules on the exterior outside the egg. But that's not actually the case, and that's a good thing. The developing embryo needs oxygen, just like we do, so the shell needs to be permeable. This allows oxygen to diffuse into the egg and then the waste carbon dioxide to diffuse out of the egg. But this permeability also means that other small molecules, such as water, can often move through the shell as well. So the shell is covered by a cuticle. This is a waxy coating that allows oxygen and carbon dioxide through, but largely prevents water from getting through. And just inside the shell are two membranes, the outer and inner shell membranes. These help keep the contents of the egg inside, as well as provide a small airspace to allow exchange of oxygen and carbon dioxide between the embryo and the outside world. Inside the inner shell membrane lies the albumin, or the egg white. This portion contains much of the protein and water that the embryo will use to grow into the chick. Inside the egg, the albumin is a transparent, pale yellow, and only turns white when heated or exposed to acid. Separated from the albumin by another membrane is the yellow yolk. The yolk contains lots of fat. Remember that fats store long-term energy, and it's this energy that the developing embryo will use to fuel its growth. Now, in this image, the red spot is the developing embryo that will become the chick. It sits at the interface between the yolk and the albumin. This allows it access to both the protein molecules in the albumin and the fat molecules in the yolk. Now, this embryo starts out as a quite fragile bundle of cells. It can be seriously damaged if it undergoes trauma, such as might happen if it contacts the inside of the shell, as might happen when the hen moves it about. To prevent this damage, there are two chalazae, which are bungee cord-like structures holding the yolk in the center of the egg. You can see these white strands when you crack open an egg, although they will have collapsed from the drawn-out line structure shown here into a ball-like shape. Now, when we consume an egg, we don't want to consume a developing embryo. So, to remove any fertilized eggs, we use a process called candling. This used to involve placing a candle behind the egg and looking for the small red blood spot inside. We've since moved to using more convenient light sources, but the general idea is still the same. Here's a link to a good video in which you can see a farmer candling a few eggs. I'll also include this link in the description below. So, how should we store our eggs? We want to keep them cold, still, and sealed. As the temperature is reduced, chemical reactions slow down, which then reduces the rate at which our food deteriorates. So your eggs should be stored in the refrigerator. Shaking the egg also increases the deterioration rate. Most refrigerators are manufactured with a spot on the door to store the eggs, but repeated opening and closing of the door isn't good for them. So you're really better off storing the eggs on a shelf inside the refrigerator and not on the door. Now, we also want to keep the eggs sealed. Ideally, we want to reduce moisture loss by storing the egg in an airtight container. And we also want to keep the nutritious molecules in the egg's interior from being exposed to microbes, so keep the shell intact until just before you use the contents. Salmonella is a type of bacteria that can grow inside poultry and can make humans quite sick if we consume it. Salmonella infects about 1.35 million people each year, and of those, about 26,000 are hospitalized and 400 die. Roughly 1 in 100,000 eggs is contaminated with salmonella, so the risk from the uncooked or improperly cooked eggs is relatively low, but the potential health impacts are significant. So to avoid getting sick, we need to kill all the salmonella bacteria that might be present. To do this, we can cook our egg at 140 degrees Fahrenheit for 5 minutes to get a runny yolk, or at 160 degrees Fahrenheit to get a hard yolk. Another way that we can keep ourselves safe is via a process called pasteurization, after the French scientist Louis Pasteur. Some manufacturers will pasteurize eggs in the shell, but most of the pasteurized eggs come in a carton form. Let's transition from discussing eggs to discussing meat. If we leave meat out at room temperature, it will fairly quickly become rotten. At a chemical level, both oxygen and light cause a process called oxidative rancidation. This process causes the proteins to be broken down into smaller molecules. These smaller molecules tend to be more volatile than the larger proteins, and our bodies have an instinctual negative reaction when we smell them. We evolved such a strong avoidance instinct to these rancid odors because these odors signal that the meat is not safe for us to eat. We will get very sick if we consume such rancid meat. Now, not all animals have such avoidance instincts. Some animals, such as scavengers, like vultures, have evolved bodily processes to protect themselves from the microbes present in rancid meat, and so they actually seek out such smells that we avoid. So, how can we keep our meat from undergoing oxidative rancidation? If both oxygen and light increase the rate of deterioration, then we're going to want to keep those away from our meat. So we can wrap the meat in a plastic wrap that is impermeable to oxygen. But the plastic wrap used in most supermarkets still allows the light to penetrate the plastic because they want you to be able to view the meat before you purchase it. Instead, we can also keep the meat in the dark by wrapping with a paper wrapper. And this paper wrapper is often used in butcher shops, as the customer is often able to view the meat prior to packaging, and so viewing the meat through the packaging isn't as big of a deal. We also then should keep the meat in the refrigerator if it will be used within about a week or so, or in the freezer if it's going to be stored longer. Now, while the freezer will virtually stop the rancidation process, there is a limit to how long frozen meat remains in high quality. The frozen water in the meat will eventually sublime. Now, sublimation is the process that occurs when a solid substance transitions directly to a gas phase without entering the liquid phase in between. In the case of our water, it starts out as ice and becomes water vapor in the air inside the freezer, and then that usually deposits on the cooling coils, causing ice to build up. The leftover meat has essentially become freeze-dried. Now, the freeze-dried meat is safe for us to eat, but the texture and the taste will suffer. In these two images, you can see the ice crystals form on the surface of the meat in the left image, while the wider section of the chicken breast on the right means that the portion will be tougher and chewier. So, freezing can help us keep our meat safe to eat for much longer, but it poses a significant downside. We now have to thaw out the meat. The safest way to thaw the meat is in the fridge. However, this takes a very long time because the temperature inside the fridge is only slightly higher than freezing. For a thick piece of meat, such as a frozen turkey, this process can take multiple days. We can speed the process up somewhat by placing the meat, typically wrapped in plastic, in cold water. This is faster because the thermal conductivity of water is much higher than that of air. However, the water will eventually be warmed by the air in the room, and if we don't change it out with fresh, cold water, the surface temperature of the meat can get high enough to allow microbial growth. Likewise, we want to avoid using hot or warm water to keep the surface temperature of the meat cold. We can speed up the process even more by using a microwave to directly heat the meat. However, the microwave radiation gets absorbed primarily by the outer layers of the meat, which means that the outside of the meat can get quite warm and even fully cooked, before the inside thaws. So there's still a limit to how quickly we can thaw the meat, especially if the cut of meat is particularly thick. Alright, so once we begin cooking the meat, it begins to undergo chemical reactions. At 120 degrees Fahrenheit, the red meat is rare, so it appears a dark red and is quite soft or squishy. At this temperature, myosin, one of the muscle fibers that is used to contract the muscle, begins to coagulate. That is, individual myosin filaments bind to each other. This causes the meat to become firmer as the myosin fibers can no longer slide past one another. This process also squeezes out the water molecules that were previously between the myosin filaments. By 140 degrees Fahrenheit, many of the other proteins in the cells begin to coagulate, so the meat becomes even firmer and loses more water. The meat is now a pink color on the interior. Note that in this image, the outside edges of the meat have gotten to temperatures higher than 140 degrees Fahrenheit, so they're darker brown in color. By 150 degrees Fahrenheit, the meat is medium rare and is a pale pink. At this temperature, the collagen, which helps to hold the muscle together, begins to shrink and becomes chewier. Even more water is released from the meat, so the meat shrinks even more. At 160 degrees Fahrenheit, the collagen becomes gelatin. Gelatin is the ingredient in Jell-O that gives it its wobbly-like texture. Just like Jell-O, this gelatin is very easy to separate, so the muscle fibers become easy to separate from one another. The individual fibers are still quite stiff and dry, as the majority of the water has been squeezed out from between them. But they separate relatively easily, so we often characterize this as a fall-off-the-bone tenderness. Note that the entire portion of meat is a dark brown throughout, not just at the surface. Now, because meat undergoes these changes at these temperatures, it can be relatively difficult to cook it to a high enough temperature to be safe to eat, without also heating it so high that the meat becomes dry. In fact, many of our cooking techniques applied to meat are designed to counteract this tendency towards dryness. Things like coating in a barbecue sauce, marinating, and so on. To kill all the bacteria present, we need to cook meat to 160 degrees Fahrenheit or higher. Any meat cooked to an interior temperature lower than this can still leave bacteria alive on the inside. However, if we have a solid slab of meat, such as a steak, we can cook the surface to 160 degrees Fahrenheit to kill off any bacteria on the surface, while cooking the interior to a lower temperature. This works because the animal, prior to being slaughtered, had an immune system that, ideally, killed any microbes, preventing the interior of the meat from being contaminated. Only those portions of the meat exposed in the butchering process to knives and other surfaces should have bacteria present. That is, only the surface of the meat should be contaminated. By heating these portions to 160 degrees Fahrenheit, we should then kill any bacteria introduced in the butchering and handling processes. Note, however, that this isn't a guarantee that the interior is in fact safe to eat. If the surface is broken, such as might happen if a knife slides into the meat without cutting all the way through, and therefore exposing the entire surface, the newly created cut would harbor some bacterial growth that would be hidden on the inside. Likewise, it's also possible that the animal's immune system did not successfully kill any microbes present prior to slaughter. In practice, most meat cooked in this way will not cause illness in the consumer, but those with compromised immune systems may be at increased risk. Unlike a solid slab of meat, however, if the meat is ground, then the surface of the meat has been distributed throughout the entire bulk of the meat, and this means that any microbial contamination on the surface has also been distributed throughout the bulk of the entire meat. So we cannot safely cook a hamburger to 160 degrees on the outside while the interior remains pink or red. Here's a listing of the minimum safe temperatures for a variety of different meats. Note that this is the internal temperature, the temperature of the coldest portion of the meat. The surface temperature will usually be higher than this. Once we've cooked a food to kill off any microbes, we can't simply return it to room temperature and expect it to remain safe forever. Once the temperature drops below 140 degrees Fahrenheit, microbes can begin to grow again. These microbes might be present in the form of inactive spores prior to the cooking process that survived the cooking process, or they may be introduced after the temperature drops below 140 degrees Fahrenheit, such as via the air or via the introduction of serving utensils. So to keep our food safe, we should either keep the temperature above 140 degrees Fahrenheit or below 40 degrees Fahrenheit. That is, we should either keep the food hot or we should refrigerate it. Food left in the danger zone between 40 and 140 degrees Fahrenheit for over 2 hours is no longer safe to eat. Many cases of food poisoning occur during barbecues or similar occasions on warm days in which food sits out for multiple hours at warm, but not hot, temperatures. Foods that have not been cooked or have portions that have not been cooked, such as potato salad, and especially those including uncooked egg products such as mayonnaise, are particularly susceptible. Now, foods that have already been cooked should be kept separate from uncooked foods to prevent cross-contamination. This means that you should store uncooked meats that may leak juices as low as possible within the refrigerator to avoid contaminating other foods via dripping juices. Ideally, the salad drawer wouldn't be at the bottom of the fridge, but it's impractical to place a drawer at the top of the refrigerator because it would often be above our head and thus difficult to use. If you store raw meat above your salad drawer, you should therefore use an absorbent towel or paper towel to reduce the likelihood of contamination of food inside the drawer. Now, many produce items are coated with a waxy coating for sale in supermarkets. This coating reduces the loss of moisture and reduces the likelihood of surface microbial growth. The coating also increases the shininess of the produce, which then leads to increased sales. The wax is safe to eat and will largely pass through our digestive system unchanged, so it's not necessary to remove prior to consumption. Although removing the waxy coating just prior to consumption won't harm anything either. The best practice is to wash your produce just before cooking or consuming. Washing earlier, such as immediately after you purchase it and bring it home, can cause the surface to remain wet and encourage microbial growth. Removal of the waxy coating can also increase the rate of dehydration. Note that where the cells in meat die fairly quickly after the animal is slaughtered, the cells in produce are typically still alive for weeks after harvest. Because these plant cells are still alive, the cellular respiration and metabolism are still occurring. This means that the nutrients present at harvest continue to be used up by the still living cells, which will reduce the nutritional value and may create relatively tough, fibrous tissue. While the quality of most living produce will steadily decrease over time, plant parts that evolve to hibernate, such as onions, potatoes, and other root vegetables, can enter a dormant state in which the deterioration is slowed dramatically, and such produce can remain edible for months if stored under favorable conditions. Again, because they're still alive, fruits will continue to ripen after being harvested from the plant. They'll continue to ripen, run out of sugar, and will eventually begin to decay. The enzymes that worked to ripen the plant will still be present and will continue to work away at random, slowly eating away at the tissue. This over-ripening causes a rather mushy fruit that usually tastes quite sour due to the dramatic decrease in sugar content. As we cut into produce, we do two things. First, we introduce the previously protected, nutrient-rich interior to microbial contamination. Second, we damage some of the cells as we cut them open. The molecules in the interior of those cells spill out, reacting with each other and with neighboring cells. The neighboring cells sense the death of a nearby cell. Now, animals can respond to an attack using the flight or fight response. We either run away or defend ourselves using claws, teeth, and so on. But plants don't have either of these two options. Instead, they use chemical warfare to either kill invaders such as microbes or to dissuade animals from eating that portion or the entirety of the plant using taste, or in extreme circumstances, toxins. The browning of the flesh of a cut apple shows the result of this chemical warfare. Apples have evolved to produce defensive chemicals that are released upon the death of a cell, such as when the apple is cut open. These defensive chemicals react with the cellular membranes of microbes, damaging them and reducing microbial growth. But those defensive chemicals are also indiscriminate, and so they also act upon the nearby plant cells, turning the nearby flesh soft and brown. Alright, so how should we store our produce? Because those cells are still alive, we want to keep most produce cold to reduce the cellular metabolism. We want to minimize moisture loss, but we can't simply enclose it in an airtight container because the cellular respiration will require oxygen to be present and will produce both carbon dioxide and water vapor. So we want to keep it in a humid environment while still allowing air exchange. But if the surface becomes wet, microbial growth is likely. So we also want to prevent surface contamination using a paper towel or paper bag to line the container. Note that in addition to producing carbon dioxide and water vapor, many plants will also produce ethylene gas. The ethylene is a plant hormone that speeds the ripening process. While this may be desirable if you want to quickly ripen a fruit, once the fruit is ripe, the presence of the ethylene will quickly cause the fruit to over-ripen and decay. So we want to provide enough air exchange to remove this ethylene gas. Note that this ethylene gas is also the cause of the saying, "one rotten apple spoils the whole barrel." A rotten apple produces a large amount of ethylene gas, and this ethylene then causes the remainder of the apples to ripen much faster than they normally would, which causes them all to release even more ethylene gas. And this positive feedback loop quickly causes the entire barrel to become overripe and rotten. Note that there are some exceptions to placing produce in the fridge. For fruits that evolved in tropical climates, the plants are not naturally exposed to temperatures as cold as those in refrigerators, and so they never evolved the ability to gracefully deal with those cold temperatures. For example, a banana placed in the refrigerator will stop the ripening process and the peel will turn black. Now, you can use this to your advantage if you allow the banana to sit at room temperature until it has reached the ripeness you prefer, and then place the banana into the refrigerator. The peel will still turn black, but the remainder of the banana will remain safe to eat. Similarly, avocados placed into the refrigerator will have their cellular machinery damaged and will stop the ripening process entirely. So if you place an unripe avocado into the refrigerator, it will not ever ripen, even if it's later removed from the refrigerator and returns to room temperature. Remember that meats are primarily composed of proteins which denature and change shape starting at temperatures around 140 degrees Fahrenheit. This means that cooking meats is a delicate balance between safety and taste. On the other hand, plants are primarily composed of carbohydrates, which don't denature, even at the boiling point of water 212 degrees Fahrenheit. As a result, cooking plants is far less temperature sensitive, and so we typically cook plants by boiling them for a period of time. That is, we use the boiling point of water to control the temperature of the food, and we vary the time that we keep them at that temperature. As long as there's a significant amount of water present, the temperature of the water won't get above 212 degrees Fahrenheit. Alright, so to summarize food safety, we want to keep our food cold or hot, but not in the warm danger zone between 40 degrees and 140 degrees Fahrenheit. We want to keep our food covered and dark to reduce microbial growth, oxidative rancidation, and moisture loss. We want to thaw frozen foods slowly, never allowing any portion of the food to get above 40 degrees Fahrenheit. We want to wash produce just before consumption, and we want to cook our meats and eggs to the proper temperatures to reduce the likelihood of food poisoning. Again, each week you'll have a variety of assignments due at the end of the week. First, you should read through the textbook and any additional documents I've posted on that week's tab on Blackboard. Once you're ready, you should then take the quiz. And for this first week, you should also upload to Blackboard the signed syllabus and food safety contracts, which can be found at the end of the syllabus. And if you have any questions, please send me an email.